Midterm 1 review. So I already started this review a little bit. I'm just going to reiterate some of the things that I said before. So midterm 1 is on Friday, of course. It is at 4 o'clock. Today is Wednesday, so that's 2 days from now. And it's going to be on Moodle using the lockdown browser. And so some things about the lockdown browser that I told you before is that you are not to be treating this as an open book exam. This is a system that proctors you, and it's going to monitor you by using your webcam on your computer. So treat this as an in-person exam, and what you need to do is basically log in and use your usual browser. I suggest you shut everything off on your computer first and reboot it just to make sure everything is running smoothly. And then you're going to log in through Chrome or whatever your usual browser is, and do the test. And it's going to load up the lockdown browser. So I had sent out an email on Monday about doing this sample quiz. I think only about 4 students have done this. It's very important that you do this and make sure your computer works properly with this. I'm not going to have time on Friday to be figuring out your technological problems. So it's important that you try out the sample quiz now. It's going to bring you through the lockdown browser process, so you have an idea of what that is going to look like. You're not going to get extra time if your lockdown browser doesn't work or if you have technology issues. So it's important that we deal with them now. If you do have a problem during the test, you can reboot your computer and email me or phone me. My plan is to be in my office on Friday afternoon so that you can call me directly to my office, and that my phone number is an email that I sent. Normally, it's going to ask you to show your ID. You don't need to show your ID for this, this test. If it was a final exam, I would ask you to try the test. I sent out a reminder on Monday. I will send out another reminder today, and about those who have not done the sample test. Okay, so let's talk about the test. This is going to be the breakdown, or at least similar to the breakdown. I haven't written the test yet. Sometimes a mark or two is swapped in another direction, like there could be 11 multiple choice and 4 true and false or something like that. But pretty much this is the usual breakdown. I might make the long answer a little shorter. I haven't quite decided yet, but generally the exams do follow this kind of format. You can take a look at the sample exam that I have on Moodle, and you can see the format from a previous semester. That one's about 5 years old. So the emphasis is always a little different every semester. So just keep that in mind, that some things on that test may be not relevant to what we've talked about. The things that we've talked about are not going to be relevant to that test. You do need to focus on what we covered over the semester, of course. We have covered 7 topics. So hopefully everyone has started studying is deep into this, because we've covered a lot of information, quite a bit we've covered. And you do need to know it well if you want to do well on the test. Talked a little bit about studying, and I told you that make sure you check out the sample midterm and the practice questions on Moodle, the practice quiz questions, because that is a big part of studying, is testing yourself and really making sure you can refine and understand your knowledge. Some people have been emailing me questions. That's great. If you have any questions, do email me, or you can always set up an appointment for an office hour kind of thing, and I'd be happy to answer any questions. Pretty much you need to go through and do the work. So what I want to do today is I'm going to not show you this video again, but what I want to do is go through the topics a little bit and cover some of the material. So let's talk a little bit about how to answer questions. I don't think I covered this here last day. So how to answer multiple choice questions. There's lots of tips on the internet. Check them out. If multiple choice is not your thing, definitely check them out. There's a really good article in Maclean's. I have it linked there. You can check it out, and it gives you some strategies. So one thing to not do is don't guess C for everything, or don't just guess the longest answer. Those are really not good strategies. Number one, read the entire question. and then answer it in your mind before looking at the options. That's a pretty good tip because usually then you have an idea of which correct answer to look for. Always pay attention to keywords that may be important as well. Eliminate wrong answers. Usually you can get rid of 2 or 3, if you're lucky, 4 of them before you find the correct answer, and then select the best answer. Generally, I think there's a little bit of a disagreement on this, but I think that often go with your gut. Some people often will go back and change the answer and then they change it incorrect. Again, some disagreement on that. Some people, maybe a little bit of thought can help you with it. Make sure you answer all the questions. At least you can do is guess, and you might even get lucky, right? But do read the questions carefully. Like I said, there's lots of tips on the internet for answering multiple choice questions, and I encourage you to take a look at them if you're not as good at them as some people. Answer the ones you know and go back to the ones you're unsure of. On Moodle, there is a way to add a flag to the questions you want to go back to. So it's important you answer the whole test. Don't get stuck on a question and waste 10 minutes and then you realize, your exam is halfway done. Answer the things you know, go with what you know first, and then go back and try to fix the ones that you don't. So what about written questions? Here's some tips for some written questions. Always take a look at keywords, okay? Every written question is going to have some keywords, and it's important that you address what those keywords are, okay? Make sure that you're not just, oh, it's about membranes, and then you go off and then you come later and realize the question was about membrane transport, okay? Make sure that, in that case, transport will be a keyword. Answer the question directly, clearly, and precisely. So make sure you answer the question. This is one of the biggest problems I have on my tests, is sometimes people don't answer the exact question. Obviously, you need to know the information, but answering the question directly is important. Another one is look at how many marks the question is worth. If the question is worth 1 mark versus 5 marks, then you should be given an appropriate response. So for my test, 1 mark is roughly 1 sentence. Now you don't need to answer things in sentence form. You can use point form, but 1 sentence doesn't mean like 4 words, okay? A sentence is 1 big complete thought. And so your answer actually may be 2 sentences. It may be 1, but that's kind of what I'm looking for, right? So think of a 1 mark question. If I'm gonna mark things in half mark increments, then I'm probably looking for at least 2 points, or a point in an example or something like that. That's my next point. Use examples, always use examples. If I ask you to define what, let's say a macromolecule was, right? So a macromolecule is a large molecule. Usually they're formed of smaller monomers. Your definition would maybe talk about those kind of things, but an example, a protein, a lipid, a carbohydrate, nucleic acid, that's a macromolecule. Be organized. If your answer is a mess, whether it's bad handwriting or I'm gonna have to read it 17 times in order to understand what you're talking about, you are not gonna get full marks. Even if you have everything there, if I have to read your answer several times, I'm going to dock you marks for being less organized. The last thing you wanna do is annoy the person who's grading your exam. So being organized is helpful. Okay, so that's kind of it for my tips, and I'm hoping that we can go through these and we can maybe give you some more tips as we go along, but some things to think about. So let's talk about the topics that we went through and kind of give you a refresher course on what we're looking at, and I think I have a few sample questions along the way. So question 1 was an introduction to the cell. So we talked a little bit about what cells are and what life is, and a little bit about the history of cells, so Robert Hooke and Van Leeuwenhoek and cell theory and those kind of things. We talked about classification of living things. So we talked about taxonomy and phylogeny, for example. We talked about nomenclature of organisms. And then we finished off this topic looking at the scientific method. And the scientific method, we cover a lot of it more in the lab, but be prepared to answer at least, 1 or 2 multiple choice questions on it, in terms of asking you things like, what is an independent variable versus a dependent variable or something like that. So I think I have a sample multiple choice question here. In fact, it is about the scientific method. So it says here, in a scientific experiment, the independent variable. So this, by the way, for a multiple choice question is the key word. independent variable. Okay, so now we got to look at the questions, and I'll go through the process of eliminating the ones that I know are incorrect. Okay, so a variable is not the control. That's an easy one I can eliminate. Excellent. Okay, has no impact on the results, is not altered, is something which changes observed, something which is deliberately manipulated. Okay, so I have to think about these a little bit. And of course, if you don't know what an independent variable is, that's a problem. You need to know what it is. So there's dependent and independent. So dependent is the responding variable. So that means the independent variable is the manipulated variable. So that is useful information. And if this was paper, I would encourage you to write all over your test. Unfortunately, not on paper, we can't do that. But let's take a look here, okay? Deliberately manipulated. Okay, that sounds right. Let's just check the other ones. Changes observed? No, that would be dependent. Is not altered? No, of course. Variables are always altered. Has no impact on the results? Of course not. So A is the correct answer. Okay, so once I was able to make the connection that the independent was the manipulated variable, then it was pretty easy for me to narrow down the selections. True or false? So true or false, by the way, multiple choice or 1 mark each. True or false are a little simpler, so I usually make them half a mark each. Again, read the question carefully. And again, if you haven't studied, you're going to have a hard time answering these things, right? So Antony van Leeuwenhoek, hopefully you remember who he was. He had those little microscopes that kind of look like this, right? Look like a paddle with an arm on it, and there's a little lens on it. Was the first person to observe cells using a microscope. So the answer is false. Turns out Robert Hooke was the first. Remember, he looked at cork cells. So he was the first person to look at cells. Van Leeuwenhoek was probably the first to observe bacteria, though. At least that we know of in terms of recorded history. There's possibly others before him. All right, moving on. Topic 2. Topic 2 is all about the macromolecules. So the lipids, the carbohydrates, the amino acids, and the nucleic acids. So it's important that you are able to distinguish between the different types of molecules. And I think I have a practice question here coming up. So I am going to have some short answer questions on the test. And so some of them might be definition type questions. That's traditionally what I used to ask, is a lot of definition type questions. I've been trying to get away from those and ask other questions too, but be prepared to answer definition type question. So macromolecules, it says define polymer. So I may have shown you this exact question before as a sample midterm question. But like I said, this is worth 1 mark. So I want to show you a sample written question, right? So 1 mark, a large molecule or macromolecule composed of repeating subunits. Okay, so I think that definition was directly from the glossary of the textbook. And you can see I kind of have 2 pieces to it. Half a mark for saying something about a molecule, large molecule. Half a mark for saying something about you've got a bunch of subunits. And if you don't quite have all that, give me an example, right? I'm not going to give you a bonus mark for having an example, but if you're missing something in the actual definition, the example is a place where you can get those extra marks and get the 1 out of 1 on it. Okay, so don't treat this as trivial because it's worth 1 mark. If you're doing 5 of these and you get 0 out of 5, that's a good chunk of the test, right? Okay, so here's another question. It says, like I said, I'm going to have a question on the exam. Previously, I've had matching type questions or multiple choice type questions around these things, or fill in the blank, right? So this is basically asking us to classify them as a sugar, so carbohydrate, a fatty acid, so a lipid, an amino acid, which is part of proteins, a nitrogenous base, which is part of nucleic acids, or a sterol, a steroid, which is, of course, a lipid as well. So let's take a look at these. Probably the easiest one in my mind is this one here. This is a sterol. So remember, it's got the 4 rings. So 1, 2, 3, 4. In fact, this one here is cholesterol. Okay, what else do we have here? A sugar. Okay, so remember, sugars have lots of alcohols on them, OHs. Okay, so maybe I'll put a little note about that. And often they're ring structures. So let's take a look at that and see what fits that bill. So we have two ring structures at the top. The one on the left does not have any OHs on it, but the one on the right does. And so this one here is a sugar. In fact, this one here is actually glucose. Notice we have some other ones with OHs on them. This one has a single OH, not enough to make it a sugar. This one has a single OH, not enough to make it what I call sugar. Usually sugar have multi-alcohols on them. And in fact, this one here, we could technically call it a sugar. It's kind of in a gray zone. Technically, biochemically, we call it a sugar derivative, but for the purposes of this course, it technically is a sugar. This is actually glycerol, by the way. You can look that one up. And some people classify it as sugar, some people classify it as a sugar derivative. Okay, fatty acids. Remember, fatty acids have a whole bunch of carbons and hydrogens, right? And that's pretty much, they're made of almost all that. And that is exactly what this one here is. And that one, I believe, is palmitic acid, but I can't remember which one that one is. Okay, so what do we have left? We have an amino acid and we have a steroid. Oh, no, we did the steroid already. An amino acid and a nitrogenous base. So remember, a nitrogenous base has lots of nitrogens in it. Both of these compounds that are left over have nitrogens in it, but this one has lots, and it's in a ring structure. So I'm pretty sure this is my nitrogenous base. And that, in fact, is, it looks like it's cytosine, I believe. I'm not 100% sure about that. I'll have to look that one up. And the last one is an amino acid. So this has an amino group and an acid group. And so that's an amino acid. So there are some practice questions regarding this on Moodle in the quiz section. So take a look at that, and hopefully you can do well on this. I think I have another one here. So just sort of bigger structures in this case here, and we can take a look at this one as well. So nucleic acid, again, I'm looking for the nitrogenous base, right? And that one there is adenine, but this time it has the sugar and the phosphates on it. So that's a nucleic acid. I'm just going to put Na for nucleic acid. What else do we have here? Phospholipid. And if we're lucky, actually in this case here, it looks like things are in order. Notice there's a phosphate group there. This little thing here is the phosphate group. You see phosphate groups on nucleic acids as well, but that's a nucleic acid. This one here is a lipid because of all these carbons and hydrogens. So that's my phospholipid. All right, triacylglycerol. So remember, triacylglycerols kind of look like this. You've got a glycerol, and you've got 3 fatty acids. So these are fatty acid 1, 2, and 3. I don't see that structure. So this one's not even here. What about a protein? Okay, do we have any proteins here? So this structure over here, remember these cartoon structures where you see these ribbons and these arrows, those are usually representing proteins. What about carbohydrate? Yes, we have a carbohydrate here. In fact, this is cellulose. So this is a carb. And this one here is cellulose. Remember, I'm just looking for all of those OHs, tons and tons of OHs there. It's usually an indicator of a carbohydrate. And that's what it looks like here. And one more we haven't looked at, and I'm hoping for everyone this is obvious, but this is DNA. And DNA, of course, is a nucleic acid. Okay, so there's going to be a question, like I said, matching or fill in the blank or a couple multiple choice or something. So it's important that you are able to distinguish the differences between the macromolecules. All right, then we looked at viruses, the SARS virus and COVID-19. Okay, so it's really important that you know what these things are. This here, SARS-CoV-2, right there, that's the name of the virus. COVID-19, that's the disease. So maybe that's the first little thing to know, is that there is a difference between those two things, right? They were named by independent committees, whether you like them or not, that's what they are. So it's important that you know what those things are, okay? And so take a look at this unit. And the first part is about viruses. So a little bit about what they are, what they're made of, and what they do. That's the first thing to learn. The second part is to learn a little bit about the coronaviruses. And so there's some general coronaviruses, and there's the one that is specific to this pandemic. And then lastly, a little bit about the disease. And there's one other section which is on vaccines. So maybe I'll write a note on that. Bus vaccines. So I think last semester I had a matching question about the different types of vaccines. So make sure you review those as well. So here's a couple of sample questions. So here's a tough question, right? What type of genome does SARS-CoV-2 have? So just keep that in mind. Some questions are going to be easier than others. And I am always going to put in every one of my exams, one or two really difficult ones to really reward the people that studied hard. Some people might get an A or an A+, and I want to reward you for studying hard for that. So looking at this, again, this is really tough. If you don't know anything about this particular virus, this is probably the only one that you can eliminate. Because we know viruses have RNA or DNA genomes. This is in fact an RNA virus. And that's actually the correct answer there. But again, that's something you'd have to really study to know. Here's another question about a vaccine. It says a vaccine is produced by using a weakened version of the pathogen. What kind of vaccine is this? Okay, so this is really the key word, a weakened vaccine. And if you know what all of these words mean, the obvious answer is this one here, attenuated. Weakened or attenuated technically means weakened. We're probably looking at those vaccines, all those other types. Again, you're going to have to study what they are in order to do well on that question. There's the correct answer. Okay, moving along. Topic 3 was on mostly microscopy and a few other kind of miscellaneous things around studying cells. So you need to know about these different types. We talked about light microscopy right here. We talked about electron microscopy, and we talked about scanning probe microscopy. And each of these had some different techniques that we discussed. Here's another short answer question. Define compound microscope. So anything like this, you've got two words now that you have to address, right? So here's an answer. So a microscope is a device used to magnify an object, and maybe you would talk about lenses or something like that when discussing what a microscope is. But the compound part you have to address, and a compound microscope, of course, has two lenses in order to magnify the object a little extra. So there's kind of what a complete definition looks like. So I wanted to show you that one as kind of a different type of question. True or false? There's the answer. Some types of SPM, scanning probe microscopy, can visualize atoms. And that was something that we had highlighted as a special feature of probe microscopy, is it can look at very, very, very small things, in some cases even atoms. Something I had talked about in lecture was that this is basically a previous type of long answer question that I've given before. So remember, this will be worth 5 marks. And so you gotta be discussing at least 5 things. Okay, and so this is talking about a light microscope and electron microscope. So you might want to make a table, right? You could have light microscopy maybe here, and you could have electron microscopy maybe here, and then over here you could discuss the different types of features. So you could say maybe the first feature you're going to look at is illumination source. Okay, so for light microscope, you could just say light, and that would be okay. But we want to be thorough. We want to make sure you're going to get the 5 marks, right? So you could actually give me more details. You could talk about natural light. You could talk about light bulbs. We could talk about lasers, right? Those are all examples, right? That's going to help to give me lots of information. Electron microscopes, I didn't tell you really anything about the different types of electron beams, so we're just going to say electron beam, unless you know more about electron beams. Okay, and so that would be worth 1 out of your 5 marks. Okay, if you're skimpy on details, I might not give you a full 1. I'll give you a 0.5 or a 0.75 kind of thing. And now you got to discuss a bunch of other things. So notice it's actually asking you to give examples. So if you don't give examples, you're not going to get 5 marks, even if you give me an amazing answer. And it actually tells me to discuss SEM and TEM, so the 2 types of electron microscopy. So some of the details in there are going to cover that as well. So make sure you give me lots of information and you'll get your 5 marks. Number 4, we talked about membranes, and this was kind of 3 lectures or 3 mini lectures, depending on how you want to look at it. If you think about this, we talked about first membrane structure. Membrane structure. 2, we talked about membrane transport. And 3, we talked about cell walls. So all of those things you need to make sure you learn a little bit about. Okay, membrane structure included these things, right? Phospholipids, proteins, and carbohydrates. Make sure you know what the functions of those things are. Transport, we talked about different types of transport. We talked about passive and active types of transport and bulk transport. And cell walls, we talked about plants, animals, bacteria, and fungi, and a few other things. But those 4 things are things to know. And obviously, animal cells don't have a cell wall, but they have an extracellular matrix. Here's a sample question. Which of the following molecules cannot? So again, there's a key word. I don't always bold them or capitalize them, but do read it and make sure you understand the key word. So which one is false for going across the plasma membrane? Okay. Charged molecules, small molecules, hydrophobic, uncharged, non-polar. So remember, the plasma membrane is hydrophobic, okay? Or at least the lipid layer is, so hydrophobic. So small things are good, hydrophobic things are good, uncharged things, non-polar. So charged, right? So charged is kind of the opposite of hydrophobic. And so it's not compatible with the environment inside the membrane. All right, so I wanted to just kind of compare these things a little bit. Okay, sometimes students get a little mixed up between cell membranes, cell walls, and extracellular matrices. So let's fill in this table and talk a little bit about what these things are and what they're made of. Okay, so cell membranes, what are they made of? They are made of phospholipids. They are made out of proteins, and those are usually about 50-50. And sometimes there's a small percentage made out of carbohydrates. And those carbohydrates are in the form of glycoproteins and glycolipids. Okay, so what is a cell wall made of? Well, it depends on what organism you are. Just remember, plants are made out of cellulose. Bacteria are made out of peptidoglycan. And fungi, their cell walls are made out of chitin. And remember, for bacteria, we had the gram-positive and the gram-negative. So there's kind of 4 cell walls to learn a little bit about. Notice there's no protists or archaea there. You can learn that information, but it's not as important as those 3. Extracellular matrices, we have, this is in animal cells only. Animal cells only. And in this case, it depends on the tissue, but the example that we gave was connective tissue, which is mostly collagen. Bone tissue, it's, what example? Bone tissue is, of course, usually mineralized calcium minerals of some sort. Okay, so back to membranes. What kind of cells have these structures? All cells have membranes. In this case here, what kind of cells have cell walls? I guess we've already answered that question. Plants, bacteria, and fungi, archaea, and some protists, and extracellular matrix, we're talking about animal cells. And we'll say some animal cells. What is the function of the membrane? Here's where it gets a little more murky. And the thing is, maybe not necessarily murky, but complex. Membranes have many functions, right? The main one is obviously to contain the cell. And the other main one is to transport materials in and out. Transport materials in and out. And there are many other things we could go on about the membrane, some which we're going to cover in this course. We could talk about nerve transmission. We could talk about energy production in the mitochondria. We could talk about reception of signals from hormones. We could talk about enzymes that are on membranes and so on. But those are kind of the big two, right? Containing the cell's contents, I guess you could say, and transporting materials in and out. Those are kind of the big ones addressed in this unit. What's the function of a cell wall? So it's a hard structure, right? So we're talking about protection, and also it will give the cell its shape. Okay, so that's a very different function of the membrane. Remember, it's outside of the membrane, and so it's protecting the membrane as well. The extracellular matrix, it kind of depends on what you're talking about in terms of, connective tissue, for example, is holding cells together. Bone is to give us hard structures so we can move around. So I'm just going to kind of say various. And this is the kind of thing that you'll get expanded on if you take more cell biology classes. You're going to get into all these types of examples and the huge variety of things that different types of extracellular matrices can do. All right, so time check. I think we're doing just fine for time. A few more things to look at here. Question 5 is, of course, the prokaryotic cells. So don't forget, we discussed the differences between bacteria and archaea. Okay, we talked about archaea a little bit, so be prepared to answer at least a little bit of the information about archaea on the test. So let's take a look. We've got this little image here, and it has some different bacterial structures. Let's take a look at these things and see if we can remember what they are. So there's fimbriae, which are also known as pili. And these are, I'm going to put a P for protein, made out of protein, and they're for attachment. All right, number 2 is a ribosome. I know it's not very well labeled. I did not label it myself because it looks a lot like the cytoplasm. But ribosomes are made out of protein and RNA, and these are for protein synthesis. Okay, I'm not going to make these notes very thorough here because we made a lot of notes already. All right, what's number 3? Number 3 is the DNA. And remember, we have a name for that. And it is the nucleoid, which is not a membrane-bound structure. It is a loose structure, has the chromosome in it. Inclusion, you can see is the next one. That's basically a food particle. We didn't really talk about those. What's number 4? Number 4 is a plasmid. And plasmids are also DNA. They're kind of like mini chromosomes. And sometimes they carry drug resistance and things like that. Number 5, what is that? It is a flagellum. Flagella is many. And this is also made out of protein. And this is for swimming or motility. Okay, so number 6, 7, and 8 are a little bit harder to distinguish what they are in terms of, they're all parts of the layer of the cell. Now the skinny one, number 6, that is the plasma membrane. We just talked about membranes. Number 7, that's the cell wall, and that one is made out of peptidoglycans, so PG, we'll say. And that's cell shape and support. And then one more. What is that one outside of the cell wall? That is the capsule. So remember, we talked about these as a structure called glycocalyx. And that was capsules and slime layers. And these things are usually for attachment as well. And they are glyco, which means they're made out of carbohydrates. So I'll put a C there for carbohydrate. Okay, so this is not meant to be going through everything in detail. I know it's a bit of a whirlwind tour, but I'm hoping this is going to prime your memory for some things, or at least inspire you to learn some of the things that you still need to cover in your studying. Here's some short answer questions. Define amphipathic. Okay, what was that again? So let's take a look at the definition. This says describes a chemical compound that is both hydrophobic and hydrophilic. So this is actually relevant to a previous topic, not the bacteria topic, but this is actually relevant to the topic on membranes because an example of this is a phospholipid. Okay, and what about the next one? Endospore. What was that? So an endospore, you can see the definition. It's a thick coated dormant structure, and the coat is usually made of peptidoglycan, and it's made by some bacteria when they experience shortage of nutrients such as water or food or something like that. And it's a dormant, kind of tough structure, right? All right, topic number 6. Let's take a look at this and see if we can identify the features on this one. So number 1, hopefully that one's easy. That is the nucleus, okay? I'm probably not going to write a lot of notes on this one here, depending on the space. I'm not going to write notes on the nucleus. Remember the nucleus, and then there's that other thing inside the nucleus which stains really well, and that is the nucleolus. So don't get the two mixed up. So the nucleolus, remember this is for ribosome assembly. Ribosome assembly. All right, so what about number 3 and 4? So one of them is attached to the nucleus and one of them is kind of a little bit more exterior. So if you guess Golgi and endoplasmic reticulum, then you are correct. And remember the endoplasmic reticulum, there is the rough ER and we have the smooth ER. So 2 different types of endoplasmic reticulum. The rough is for protein synthesis, the smooth is for lipid synthesis. So they have different functions. You don't really see a distinguishment between the two on this diagram because it's a cartoon, it's an artist. The Golgi is for protein sorting and processing. And it's going to basically package these things up and send them to the right location in the cell. Right, number 5 is a little bean-like structure, and if you guessed mitochondria, you are correct. So that is the site of cellular respiration. So actin filaments down here, we're going to talk about that in topic 7. So we'll skip that for now. Number 6 is the plasma membrane. You can also call it the cell membrane. Both terms are correct. So number 7 and 9, it's hard to tell what they are. They're just little blobs. But those things are actually representing the peroxisomes and the lysosomes. Peroxisomes are oxidative compartments, and they can break things down, such as weird fatty acids. Lysosomes are also digestive compartments. They're acidic, and they can break down other food as well. The cytoplasm is listed on there, and another component of the cytoskeleton, which will be topic 7. So you don't need to worry about that until after the midterm. That will be next week. And there's all the answers. Okay, fill in the blank kind of questions here. Let's take a look. Which organelle do each of these functions? So translation, of course, again, here's a case where you need to know what that means, right? Translation is protein synthesis, okay? But notice there's a key word here, membrane proteins, okay? So we're talking about ribosomes, of course. And I have all these questions here. Do I have answers here? I have to write them in my pen. So we're talking about ribosomes, which do translation, but this membrane protein part, right? So this is ribosomes on the rough ER. Okay, so a specific kind of ribosome, not just the loose cytoplasmic ones. What about synthesis of cytoplasmic proteins? So this is kind of asking for the same thing, but now we're talking about cytoplasmic ribosomes or free ribosomes or soluble ribosomes. You can use any of those terms. Number 3, ribosome subunit assembly. We just reviewed that on the previous slide. That's the nucleolus. What is something that supports the nuclear membrane? So we haven't talked about that today, but we did talk about when we talked about the nucleus. There are these proteinaceous fibrous structures inside the nuclear membrane, and they are called nuclear lamina. And you can actually just call them lamina. That is also acceptable, but full name is the nuclear lamina because they're found in the nucleus and they're supporting the nuclear membrane. Okay, glycosylation of proteins. So remember that means adding a carbohydrate. So there's 2 places this happens, one, the rough ER and the Golgi body. Glycosylation happens in both of those locations. Number 6, breaking down old organelles. So this has got to be something that breaks things down, right? So there were 2 things. There's the peroxisomes and the lysosomes. And in this case, the correct answer is the lysosomes. There was an image there I showed you from the lectures that had a lysosome breaking down. It was actually breaking down a peroxisome and a mitochondria. And then the last one, photosynthesis, I'm hoping that one's an easy one. The answer here is chloroplasts. There we go. Okay, so hopefully that was, like I said, a whirlwind tour through some of the course material here. I have a few more things to say about the midterm. Like I said, be prepared to answer 1 or 2 long answer questions. They're all worth 5 marks. Here's another example of a long answer question I've had. There's only so many I can ask, in terms of each unit. There's really only 1 or 2 long answer type questions I can ask, unless that are kind of obvious. So here's another one, something we talked about in several of the units in terms of what are the differences between prokaryotes and eukaryotes. So I could ask the difference between bacteria, archaea, and eukaryotes, or there's a lot of combination of these kind of questions. Again, you want to discuss at least 5 points. And in this case here, the key words, right, are prokaryote and eukaryote. So you better tell me who has a nucleus. Okay. If you get that wrong, that's an essential part of this question, telling me who has a nucleus. So the answer is the eukaryotes have a nucleus, of course. What else? Some major concepts to study. So kind of looking through this, I guess I didn't throw in viruses and vaccines. I could throw that one in as well. Viruses and vaccines. But in terms of if you take a quick look at the units, these are some of the key concepts, right? We've got molecules, cell parts, so eukaryotic and prokaryotic cell parts, types of microscopy, viruses, and vaccines. Okay, so those are kind of the key things. So make sure you spend a little bit of time, maybe equal amount of time on each of these concepts, right? So if you study for 6 hours, then at least an hour on each of these. Another thing I always suggest that people do when they're studying is talk to your friends and find out what they're doing to study. How long are they studying? Are you studying 1 hour this weekend? Are you studying 18 hours this weekend? And see what other people are doing and ask them what is working for them. Because I really want people to do well, and I realize that these tests can be quite difficult. So that's why I have, and I can't emphasize this enough, do take a look at the sample midterm and the practice questions. And practice does help. It can help. What else do I have here? Actually, this is the last slide I have for you. Just a good luck balloon. So I don't have a real good luck balloon, but I found a picture of one on the internet. So I guess I zipped through that pretty fast, and we're done a few minutes early. I don't know if there's any questions. If not, you can always reach out to me during my office hours.